Hello everyone, welcome to A Vision for the Future, Episode 5. Imanis Life Sciences has created this series in order to increase awareness and utilization of new technologies that will accelerate scientific progress and provide higher quality, meaningful, and actionable data sets. You can explore our previous 4 episodes with innovators from MI Labs, Bruker, Fujifilm Visual Sonics, and the Mayo Clinic below. In this episode of A Vision for the Future, we will be hearing from Dr. Vincent Racaniello. Referred to as the world's virology professor, he has been a pillar in the field of virology for the past 40 years and is widely known for his achievement of creating the first infectious clone of an animal RNA virus in 1979. He went on to establish his own lab at Columbia University in 1982. In 2008, Dr. Racaniello started his widely popular podcast, This Week in Virology, or TWIV. Along with his work on several other podcasts, he continues to lead a lab at Columbia and has co-authored the Principles of Virology textbooks. In this conversation, Dr. Racaniello shares his thoughts on his groundbreaking work at MIT, his perspective on the way access to data has changed in science and will continue to change, what inspired him to start his podcast, and his thoughts on how oncolytic virotherapy is powering the future of cancer research. We'll begin with Dr. Racaniello's immense achievement in the year of 1979. He was working in David Baltimore's lab, who had just won the Nobel Prize.
And I had the opportunity to go to MIT in Boston to do a postdoc with David Baltimore. He had just won the Nobel Prize a few years earlier for discovering reverse transcriptase, and that gave birth to the recombinant DNA revolution. For a few years, there had been a moratorium on cloning entire viral genomes as DNA because scientists thought it might be dangerous. So when I got there, this is 1979, in the fall, the moratorium had just been lifted. So he said, I want you to make a DNA copy of poliovirus. We can sequence it. No viral genomes had yet been sequenced. It's amazing. Today, you can do it in 10 minutes. It's just remarkable how the tech has changed. He said, I want you to do the sequence of the genome and then see if it's infectious. Because maybe if you put the DNA into cells, it'll start an infectious cycle. So that's what I did.
Looking back on this momentous achievement, Dr. Racaniello did not fully recognize the implications it would have for future therapeutic development.
I knew that virologists would make infectious clones for each of their viruses, and they would use it to study them. But what I didn't appreciate was how this discovery would be used in therapeutics. And today we see enormous strides being made in oncolytic virotherapy. And that is where it's really taken. I mean, for research purposes, people have learned a lot about studying viruses using that reagent. But therapeutically, it's just been amazing. And that's going to be the future, for sure.
Imanis passionately supports oncolytic research through our encompassing product line of reporter gene lentiviral vectors, cell lines, and oncolytic viruses, and through our ongoing collaboration with industry trailblazer Virology. Dr. Racaniello comments on the role oncolytic virotherapy will play in the future of cancer research.
Huge. It's going to be huge. I say almost every cancer is probably going to be treated with some viral therapy in the future. We see a few that have been approved already, FDA approved, and they're going to get better because we're learning new things, and I think combining them with checkpoint inhibitors, immunotherapies, drug convertases, all these ideas that have just come up in the last 10 years or so, we're going to have more of those, so it's going to get better and better, it's going to get more specific, and I think it's just going to revolutionize cancer treatment. I have to tell you, when it started, I didn't believe it. Decades ago, a long time ago. It started with reoviruses, I believe. This first reovirus found to replicate in tumors preferentially. And I said, there's no way that this can work because the viruses aren't going to be specific enough. They're not going to be targetable. And in the meantime, all these developments came out. And it's great. It's a great testimony to persistence. Okay, the first time doesn't work. What about this? And then as time goes on, new developments are enabling new technologies. And that's the great thing about science. If it doesn't work today, it could work in a couple of years. Just keep at it. It's wonderful. It's a great field.
With so many exciting developments emerging every week in the field of virology, Dr. Racaniello had an idea. Why not create a podcast to discuss them? In 2008, he did just that, starting This Week in Virology, or TWIV.
So I thought, This Week in Virology, that would be so geeky, almost so geeky that no one will listen, but that's part of the coolness of it.
But they did listen. TWIV now gets over 10,000 downloads an episode, and the audience expands beyond just scientists. People of all backgrounds and ages tune in to learn about advancements in the field of virology.
That was the model that I wanted. I wanted to just talk, have casual conversations, to let people sense what it was that we did.
When I asked Dr. Racaniello what he thought the largest challenges facing the field of virology were, he comments on the importance and difficulty of getting the public on the side of science, and says this was a large reason he started the podcast in the first place.
I think a lot of people are suspicious of scientists in general. And I think that arises from not understanding what we do. For years we have been hidden, essentially, because there was no internet for many years. It was just us publishing and talking with each other, and the public didn't really hear any of those conversations. And that's why I started the podcast, in part, to get the public to hear it.
Now, as the scientific world trends more towards open access data and open access publication, Dr. Racaniello sees a powerful opportunity to open up these important scientific discussions more than they have been in the past.
Open access data, where you put your data online and you let other people see it to help them. I think this is so important. The way science has always worked, it's been somewhat secretive. People want to protect their own work. They want to get an edge on the competition. But that only slows down innovation. And it's not about you and your particular glory and your lab. It's about advancing science for everybody else. Towards the end of my career, I view myself ultimately as an educator. We do research, but I want to tell the world about it, and I think it's really important. And the feedback we get tells me that this is worth it. Teaching people about science right now is the most important thing I can do, because I can't tell you how many people have written and said, "You've turned me on to virology, you've turned me on to microbiology. I'm going to have a career in this area because of you." I tell you, there's nothing better than hearing that. As a teacher, there's nothing better that I can do.
I will have all of Dr. Racaniello's social media platforms and blog linked below, as well as some of his key papers. If you'd like to learn more about the innovations produced at Imanis Life Sciences, explore our website, where you can learn about our expansive collection of reporter gene products, as well as our custom research services. Thank you very much for watching this episode of A Vision for the Future.